Okay, so hopefully we're all plugged in and we can see. So today's lecture is a pretty interesting one because it really does kind of describe and tell you a lot about the cutting edge of molecular biology. And it's crazy because when I was revisiting these slides, I ended up having to change lots of them, even from last year. So things are progressing at such a rapid rate with sequencing technologies that it's really, really hard to keep up. And I dare say in the next 5 years that many of the things we're going to be discussing now will be obsolete. In fact, I even had to kind of cross out some slides and basically get rid of things which were supposedly cutting edge not that many years ago. Alright, so the gist of it is basically it's a very quickly developing and evolving field. So you're all very familiar with sequencing and Sanger sequencing, I hope. You've been given a lecture specifically on sequencing, I guess. So if not, we'll hopefully this little link at the bottom of this page kind of does a bit of an overview of Sanger and some of the next generation sequencing. But basically, next generation sequencing, as it was termed quite a while ago now, but it's just a massively parallel way of sequencing. So it's a way of sequencing vast amounts of sequences in as short a time as possible. Okay, so your Sanger sequencing had severe limitations. Very accurate, as you can see from your prac data. When everything is performed correctly, then the chances of having an error are very small. It's like 99.999% accurate, and any of the issues are likely induced by how the error rates of the DNA polymerases that you use to generate your templates, etc. So the main focus, I'm not going to be doing too many comparisons between Sanger sequencing, but it should be clear to you and become very obvious that the next generation sequencing is just clearly the way of the future in terms of just sheer numbers and quantity. I suppose one way of looking at that is in 2003 the Human Genome kind of project was completed or so, and that took a lot of time and exorbitant amounts of money, lots of money. By the mid-2000s, like only a couple of years later on, technologies has evolved that would have caused a 50,000 drop in the cost of that human genome sequencing. And now we're at the stage that you can actually sequence a human genome for under $1,000. So we've gone from a huge amount of money. I can't remember the exact figure, but we're talking a lot of millions to actually get that first human genome project underway or completed. And now for a measly $1,000 you can have your sequence done. And not only that, some of the machines like this Illumina, which is the company HiSec10, they can pump out so many sequences and perform so many genome runs that you can do approximately 1,800 genomes to 30 times coverage, so 30 times over a year. Right? So there are already scientific projects where people have had, where scientists have gone out and decided to sequence the genome from more than 1,000 people, and then have a look at differences in those genomes from various populations. So if you think of it, that's a massive feat that it would have almost seemed unbelievable in the early 2000s when the Human Genome Project was first underway. So based on that kind of law of or that rapid increase in technology, you can only imagine what's going to be possible in the next 5-10 years. And we will touch a little bit on some of the newer future technologies which verge on being current, and you'll soon see that some of the prospects are very exciting. Okay, so most next generation or massively parallel sequencing systems generate very short reads. Okay, so you're aware from your Sanger sequencing that you generate probably, if it's a good run, maybe a bit over 1,000 quality reads, so error-free reads that are clearly, each base is clearly distinguishable. Okay, so that allows, that's really good. However, the difference with most next generation sequencing platforms is they tend to generate very short, 35 to 700 being the longest reads. Okay, there are long read sequencing platforms, but they suffer a much higher error rate than, say, Sanger sequencing alone. So the majority and the most popular next generation sequencing platforms today are based on short read platforms. So usually 35, or probably these days we're probably looking at something like 100 to a maximum of 300 base pairs, but they're doing lots and lots and lots of them. Okay, so perhaps just to start off, we'll watch this little video which kind of puts a few things in perspective before we start entering and having a look at some of the specific technology and designs of a few of the next generation sequencing platforms. I hope I've got internet.
You need a website. Why not do it yourself? Okay. With Wix, you can create your own professional website. Really choose from
Just zoom this up. Make sure we've got HD.
Next-generation sequencing, or NGS, is a powerful platform that has enabled the sequencing of thousands to millions of DNA molecules simultaneously. This powerful tool is revolutionizing fields such as personalized medicine, genetic diseases, and clinical diagnostics by offering a high-throughput option with the capability to sequence multiple individuals at the same time. Sanger sequencing, first developed in the 1900s, is the gold standard for DNA sequencing, and it is still used today extensively for routine sequencing applications and to validate NGS data. It utilizes a high-fidelity DNA-dependent polymerase to generate a complementary copy to a single-stranded DNA template. In each reaction, a single primer, complementary to the template, initiates a DNA synthesis reaction from its 3' end. Deoxynucleotides, or simply nucleotides, are added one after the other in a template-dependent manner. Each reaction also contains a mixture of 4 dideoxynucleotides, one for each DNA base. These dideoxynucleotides resemble the DNA monomers enough to allow incorporation into the growing strand. However, they differ from natural deoxynucleotides in 2 ways. 1. They lack a 3' hydroxyl group, which is required for further DNA extension, resulting in chain termination once incorporated in the DNA molecule. And 2. Each dideoxynucleotide has a unique fluorescent dye attached to it, allowing for automatic detection of the DNA sequence. As a result, many copies of different length DNA fragments are generated in each reaction, terminated at all of the nucleotide positions of the template molecule by one of the dideoxynucleotides. The reaction mixtures are loaded on the sequencing machine, either manually onto slab gels or automatically with capillaries, and are electrophoresed to separate the DNA molecules by size. The DNA sequence is read through the fluorescent emission of the dideoxynucleotide as it flows through the gel. Modern-day Sanger sequencing instruments use capillary-based automated electrophoresis, which typically analyzes 8 to 96 sequencing reactions simultaneously.
So there's your Sanger sequencing refresher.
Next-generation sequencing systems have been introduced in the past decade that allow for massively parallel sequencing reactions.
These systems are capable of analyzing millions or even billions of sequencing reactions at the same time. Although different machines have been developed with various differing technical details, they all share some common features. 1. Sample preparation. All next-generation sequencing platforms require a library obtained either by amplification or ligation with custom adapter sequences. 2. Sequencing machines. Each library fragment is amplified on a solid surface with covalently attached DNA linkers that hybridize the library adapters. This amplification creates clusters of DNA, each originating from a single library fragment. Each cluster will act as an individual sequencing reaction. And 3. Data output. Each machine provides the raw data at the end of the sequencing run. This raw data is a collection of DNA sequences that were generated at each cluster. The differences between the different next-generation sequencing platforms lie mainly in the technical details of the sequencing reaction and can be categorized in 4 groups: pyrosequencing, sequencing by synthesis, sequencing by ligation, and ion semiconductor sequencing. In pyrosequencing, the sequencing reaction is monitored through the release of a pyrophosphate during each nucleotide incorporation. The released pyrophosphate is used in a series of chemical reactions resulting in the generation of light. Light emission is detected by a camera which records the appropriate sequence of the cluster. The sequencing proceeds by incubating one base at a time, measuring the light emission, if any, degrading the unincorporated bases, and then the addition of another base. This technology is capable of generating large read lengths, much comparable to the read length of Sanger sequencing. However, high reagent cost and high error rate over strings of 6 or more homopolymers have reduced its applications. For more details on the technical aspect of this technology, please visit our knowledge base at the link provided in the description below. Sequencing by synthesis utilizes the step-by-step incorporation of reversibly fluorescent and terminated nucleotides for DNA sequencing and is used by the W10 NGS platforms. All 4 nucleotides are added to the sequencing chip at the same time, and after nucleotide incorporation, the remaining DNA bases are washed away. The fluorescent signal is read at each cluster and recorded. Both the fluorescent molecule and the terminator group are then cleaved and washed away. This process is repeated until the sequencing reaction is complete. This system is able to overcome the disadvantages of the pyrosequencing system by only incorporating a single nucleotide at a time. However, as the sequencing reaction proceeds, the error rate of the machine also increases. This is due to incomplete removal of the fluorescent signal, which leads to higher background noise levels. Our NGS, an introduction knowledge base, provides more technical details about this technology. Sequencing by ligation is different from the other 2 methods since it does not utilize a DNA polymerase to incorporate nucleotides. Instead, it relies on 16 octamer oligonucleotide probes, each with one of 4 fluorescent dyes attached to its 5' end that are ligated to one another. Each octamer consists of 2 probe-specific bases and 6 degenerate bases. The sequencing reaction commences by binding of the primer to the adapter sequence and then hybridization of the appropriate probe. This hybridization of the probe is guided by the 2 probe-specific bases and upon annealing, is ligated to the primer sequence through a DNA ligase. Unbound oligonucleotides are washed away, then the signal is detected and recorded. After that, the fluorescent signal, along with the last 3 bases of the octamer probe, are cleaved, and then the next cycle commences. After approximately 7 cycles of ligation, the DNA strand is denatured and another sequencing primer, offset by 1 base from the previous primer, is used to repeat these steps. In total, 5 sequencing primers are used. The major disadvantage of this technology is the very short sequencing reads generated. Ion semiconductor sequencing utilizes the release of hydrogen ions during the sequencing reaction to detect the sequence of a cluster. Each cluster is located directly above a semiconductor transistor, which is capable of detecting changes in the pH of the solution. During nucleotide incorporation, a single hydrogen ion is released into the solution and it is detected by the semiconductor. The sequencing reaction itself proceeds similarly to pyrosequencing, but at a fraction of the cost. Please view our knowledge base for further details on ion semiconductor sequencing and the sequencing by ligation techniques.
In order to be able to showcase and compare the different technical aspects of each of the above technologies, the number of coverage that each run generates when sequencing the whole human, mouse, Arabidopsis thaliana, and E. coli genomes are calculated and presented here. The presented data is based on the most powerful machines of each technology. Further details can be found on our knowledge base. For whole genome sequencing data to be useful, a minimum of 30 times coverage is required. As it can be seen, the pyrosequencing method is only able to sequence the E. coli genome at enough coverage to result in valid data. The sequencing by synthesis method, which is the most popular method currently on the market, is able to generate hundreds of coverage per run. In fact, with this machine, it is possible to sequence 15 individuals within 3.5 days. The sequencing by ligation method also generates enough coverage for all genomes to be used. However, it isn't capable of generating nearly as much output as the Illumina HiSeq machines. The iron proton machine is used mostly in clinical settings because it is able to provide a sufficient size output within 2 hours. ABM offers a wide range of next-generation sequencing services.
Okay, so there's a little overview. So it even places everything into perspective when you compare it to the Sanger sequencing. Okay, so your Sanger sequencing, you're obviously aware that the main fundamental aspect of that is having the dideoxy termination reactions. Okay, so that was, I think, a pretty concise little refresher there for the Sanger at least. Now the others, next generation techniques, were probably a bit of an overload, and we'll slowly go through some of those. Now the problem here is that there's multiple techniques used and different types of strategies undertaken, so we'll try and break those down into some particular groups. That little video broke them down into a couple, but I think I will break them down into some other groupings. Okay, these slides are basically just showing you the rapid rise of massively parallel sequencing. So it's really taken off since that mid-2000s, and if you do some sort of comparison to Moore's law, which is a rate of exponential increase in semiconductor technology, well then you can see that the rate of sequencing improvements in sequencing technologies got even greater, and you can see here that the cost associated with raw megabases has just plummeted since their introduction, which is why that it's actually affordable now to go out and decide you're going to sequence 100, 200, 1,000 different humans' genomes. Okay. So if we actually break down, we'll start off with short read sequencing, which is what the majority of sequencing platforms today perform. So we're talking something like 35 to 700 base pairs, and the most popular platforms probably being 150 to 300 base pairs. What's common to all of them? Well, once you've extracted DNA that you want to sequence, you fragment it. You break it up into lots of smaller pieces. The second step, which is very common, is you ligate some adapters. So you put oligonucleotide sequences that you know the sequence of onto the ends of those fragments. Okay, a couple of different reasons for that. One, you can use them to attach to specific matching complementary sequences on substrates, and you can also use them to tag, to identify particular templates. Okay. So those two are the first two steps. And I'm not going to go into too much detail about them because they're pretty self-explanatory. The third step, which is common to pretty much all of these short read sequences, is the clonal amplification. Okay. It's only the newer third generation sequencing technologies, which we'll talk about towards the end of this lecture, that don't really involve clonal amplification. Now what do I mean about clonal amplification? Here we're talking about a method of actually using PCR to generate lots and lots of template. Okay, why do we do that? Well, most of the signals that are generated from our template wouldn't be strong enough to distinguish them from background noise unless we actually amplified them so that you had lots of them. And that clonal amplification is meaning just so you're getting lots of the one type of template amplified. Okay, so we'll talk about a couple of the different methods that are used for clonal amplification, and then we'll try and explain some of these sequencing technologies. Okay, so you saw in that little brief introductory video that most of these technologies rely upon an attachment of those DNA fragments to a solid surface before the amplification. Okay, so they've been fragmented, they've had some adapter ligated to them, so just stuck on the ends of them. That usually allows them to bind to whatever you have on a solid substrate. Okay, so you may have a bead, a very small bead, or you may have a glass surface that has a complementary sequence that can attach to the little tag that you ligated, or the adapter you ligated onto your template. Okay, so that's pretty simple. Then we do clonal amplification. Okay, hopefully that's visible there. The first method for clonal amplification is pretty much emulsion PCR. So what do we mean by emulsion PCR? Well, you start off with having lots of little beads. Okay, and on each of these beads, let's see if I can point out, as I mentioned, you have a short sequence that is complementary to the adapter that you stuck onto your template. So you may have a sample of DNA that you want to know all the different sequences of, so you tag all of that DNA with an adapter. Then when you mix it with the beads, all of your tagged templates will stick to the bead as such. Okay, they're hybridized on there. Then what happens is we actually put them into an emulsion. So everybody knows what an emulsion is. You've seen vinaigrette. So when you mix things like oil and water, you get very tiny little oil droplets in the solution. Okay, so why are we trying to do that? Well, what we can do is we can make such a fine emulsion, but we can make that emulsion out of PCR reagents basically. So we can make that emulsion, and we can make the emulsion contain a bead and all of the PCR reagents in that small little tiny droplet. Okay, so fundamentally every little droplet or missile, as we call them, in that emulsion is its own little PCR tube, if you want to look at it like that. So it has the bead and it has the PCR reagents, and the little droplet is separate from every other little droplet. Okay, you can then fundamentally just run PCR through thermocycling, and because the reagents are all in the little droplet, you get the amplification. Okay, now once you've done that, you'll end up with lots and lots of amplified product on your bead. Okay, so here you can see what's actually happening in each missile. So the template is going to be, the primer, is fundamentally what's going to be attached. Or it'll hybridize to the adapter on the bead, then the reagents will extend and you'll get a copy, and you'll end up with obviously one copy attached because it's been amplified from what's on the bead, and you'll have another that's free that then gets washed away. And after that, you're left with the extended template from the adapter that was on the bead to begin with. Is that reasonably clear, or have I done a wonderful? bead is a solid surface, so it might just be some sort of polymer. Okay, so it's not part of the emulsion as such. It's added to the emulsion as you would add the PCR reagents, and they're basically just an oil in water, and then after a process which creates all the small little oil droplets, we'll call them oil droplets, all the little missiles, they'll all have in them a bead, which is the solid substrate with the little adapters on them, and they'll also have the PCR reagents. But what you need to just realize is when the PCR process is going inside each little droplet, that what you're getting amplified is a copy of what's actually become attached to what's already on the bead. So you may have only had a couple on the bead to begin with, but then eventually, as you get a template extended and then half of it released, that other half that's released can then go and bind onto another little piece on the bead and be amplified again, till eventually you have a bead that's covered in the attached sequences. Yeah? You get it? Clear? Okay. There's plenty of little YouTube type videos which can explain this. It's just rather than watching videos, I'd like to actually talk it through. But it's not a massively difficult process to come to grips with there. You basically, if you don't want to know exactly what's going on, you're just amplifying product within a little droplet in that emulsion, and you're going to end up with a bead that has lots of clonally amplified, so lots of the same sequence attached to it. Okay. Another type, and this one's a little bit harder to visualize, I suppose, for some, is this solid phase bridge amplification. So it's no longer happening on a bead, it's happening on a solid surface. And what you get is you get your template, which is mixed in with a surface that has lots and lots of two types of adapter attached to the surface. Okay. Now you have your template come in that has previously, as we said, had an adapter stuck on it that's complementary to the two little attached oligos on the substrate. So here's one coming in. The red adapter is matched up with this red here. Then what happens? Bridge PCR happens. Okay, bridge amplification. So pretty much this will bend over, and the other end, which matches the other type of, so the blue oligo on the substrate, it'll attach to that, and then the PCR can happen, and you just get extension happening up and over. And by doing this, you end up with these clusters where lots of them have been amplified, spring back up, bend over to the next complementary pair, the PCR reaction continues, and they amplify like that until you have clusters, as we call them, of exactly the same sequence. Okay, so it's just another way of clonally amplifying that template that you've already put adapters onto. Okay, is that also equally as clear? Probably. I'm sure you'll be able to go back over and kind of view some videos, etc. if you're struggling with that. But the main concept is that you're actually getting lots and lots of clonally amplified sequences. So sequences are the same. And why? Because to generate a signal in the sequencing step, which we'll explain later, you need a strong signal to distinguish from background. So you need lots of them. Okay. So now we're going to move on from those two broad or two main forms of clonal amplification, and discuss at least two broad sequencing categories, and these are just some that I've adopted from a recent review paper. So the first one is the SBL, or sequencing by ligation, and really there's only one type of technology that's using that that type of sequencing platform at the moment, or that category, and that's solid machine, now sold by Thermo Fisher, I think, and we'll explain that a little bit later. The majority of sequencing is done by synthesis. Okay, so here we're actually using a polymerase, and we're using that polymerase in a similar fashion, I suppose, in some ways to you did in the Sanger reaction. So the first one, sequencing by ligation, there's no polymerase happening. There's just a ligase in there which attaches things. The second one, SBS, sequencing by synthesis, you're using a polymerase. Now that can be broken down into two forms as well. You've got CRT, so cyclic reversible termination, and that is all it's saying is you're using blocked nucleotides, so that have the 3' hydroxyl group with a terminator on them, very similar to the Sanger sequencing. Okay, so whenever you have that blocked 3' terminal, you can't get extension happening. Okay, it terminates then and there. And then the other form is the single nucleotide addition. Okay, so here you can the signal is generated one nucleotide at a time, and it'll become a bit clearer when we go through some methods, but each of your little clonally amplified templates is exposed to one nucleotide, one type of nucleotide at a time. So basically A's might be washed over the substrate, and if an A binds, a signal is released and detected. It's then, and we know because it's got most of the time a little terminator on it, so nothing else can, well, for the first one, nothing else can come onto it. But if an A goes over, unless there happens to be a site for another A, the polymerase isn't going to add another A. Okay, and then all the A's are washed away, and then you flood it with a T or a C, etc., or a G. Okay, and if there's complementary sequence for it, it'll attach. Okay, so that's the single nucleotide addition. Probably best explained with some examples. So we'll start off with the sequencing by ligation first. So this one, put it in there just to show you that there are some different techniques around. This type of technology is being used fewer and fewer, less and less, mainly because it's disadvantaged, as you saw if you remember from that little introductory video, that the sequencing by ligation using this method, this chemistry, only produces very short reads, like 35 bases, and there aren't too many applications for such a short read these days. So this particular form of sequencing has probably limited number of days left. I say days, but its lifetime is quite limited, I'd say. Okay. However, that doesn't mean to say it's not interesting to understand the technology behind it. And if we go through it, this is a ligase. What's actually happened is you've got an adapter here in the colored red, and you've got your template in this lighter color here, because that obviously was one of the first steps, ligating, fragmenting, and then ligating on the adapter. You then have a universal, basically, primer that can recognize the adapter, go in, the ligase will then roughly know where to go. You also flood the reaction with a bunch of these oligonucleotides that have two bases that are known, and you have some degenerate bases attached to them, and a fluorophore. Okay, so if this happened to be a T and an A, then the little sequence, the little oligo that had the A and the T will come in and bind. Right? The degenerate bases will also bind because they're mixtures, so they'll bind to anything. Okay? Then what happens is the ligase joins them up. Okay. Then what happens is you get a wash go over it, and the signal is red, and that particular AT will have a fluorophore that we can recognize, or the little camera can recognize. Then you have the product cleaved in the next step, so the fluorophore is washed away, and part of the degenerate base is washed away, so leaving 3 base gap here, and then the cycle continues over again. It's flooded with a bunch of different oligos with the 2 differing bases, and we might get, say, a G and a T bind in there, and that G and the T will have a different fluorophore, so through its round it will then give off a signal for a G and a T. Okay, and then it gets washed away, snapped off, and then washed away, and it's flooded again with those degenerate, and it goes on and on. All right, but it doesn't go for too long because we know that they're very short reads that it can give. So after about 10 rounds of doing that, the whole thing, that whole attached ligated side is cleaved, and you're left with your original template in there. Then what happens is it's flooded with a bunch of different oligos, which then go and fill in the offset, the gap. And basically after so many rounds, we end up having the same strand read multiple times, but the little known bases being added into the gaps that are left after each probe extension round. Okay. A lot of these concepts can be a little bit tricky to come to grips with the first or second time round. So trust me, if you revisit it and think carefully about it, it'll seem relatively straightforward. Okay, maybe this little video that I've got here can help. No doubt we start off with an A.
From the industry leader in DNA sequencing comes the next generation in genomics, the solid system. A revolutionary new platform with built-in scalability that delivers ultra-high throughput with unmatched accuracy. The robustness and flexibility of the solid system enables a wide variety of applications, including whole genome sequencing, targeted resequencing, gene expression, small RNA analysis, and chromatin immunoprecipitation. The workflow for each application differs only in the upfront sample prep and downstream data analysis. The sequencing chemistry and imaging steps remain constant. The solid system allows researchers 2 choices for sample preparation: fragment library or a mate paired library. In both cases, target DNA is sheared to a specific size, and adapters are ligated to the ends of the resulting fragments. A fragment library incorporates a single piece of DNA, whereas a mate paired library incorporates 2 DNA sequences that are a known distance apart in the target sample. The resulting libraries contain millions of unique molecules representing the entire target sequence. Each molecule is then clonally amplified onto beads in an emulsion PCR reaction. The sample is enriched for beads containing library template, yielding a greater than 90% positive bead count. Beads are covalently attached to a glass slide. The open slide format allows for the flexibility to analyze 1, 4, or 8 samples on a single slide. With barcoding, one can pool up to 16 libraries, further increasing the throughput to 256 samples in a single run. On the solid analyzer, the templated beads are combined with a universal sequencing primer, ligase, and a large pool of dye-based probes. The dye-based probes are fluorescently labeled with 4 dyes. Each dye represents 4 of 16 possible dye nucleotide sequences. The complementary probe hybridizes to the template sequence and is ligated. After fluorescence is measured, the dye is cleaved off, leaving a free 5' phosphate group available for further reactions. This process is repeated for 7 cycles and yields a 35 base pair read. Additional cycles can be added to extend the read length. The synthesized strand is removed, a new primer is hybridized, offset by 1 base, and the ligation cycles are repeated. This primer reset process is repeated for 5 rounds, providing a dual measurement of each base, separated by several cycles, which increases the sequencing accuracy.
All right, so if we pause that there, you can see that it all depends upon that initial primer, that initial universal primer that goes onto the DNA template. And because it is the one that's going to offset where the 2 known bases attach, because they attach and then it leaves 3, basically it leaves 3 attached that we don't know what they are, and then it attaches another 2 that we know, and you go, so you'll know every, those 2, then the gap of 3, those 2, gap of 3, those 2, gap of 3. Then it gets rid of that first strand and it puts another universal primer on, which is offset by 1, so that we can now know, we can see what the first of these 3 is. Okay, so when it goes 2 and not a 3, no 2, don't know these 3, no these 2, don't know these 3, you'll get one unique one that you didn't know before. So we didn't know this one here before, but after the second round, when we have an offset universal primer, we now know it here. Yeah. And after the third universal primer that comes in, which is offset again, we'll know what this one is. So if we compare it to the very first one where we didn't know these 2, we now know that one of it and that one of it. So we know those 2 bases there. So once you've done enough rounds, you have basically filling in the gaps. Now the beauty of this system is, because it's repeatedly reading it, the error rates are really low. It's really an accurate system because you're basically going through and repeatedly looking at the same template and reading over and over and over again. But unfortunately, it just creates those very small and short reads. All right, so we'll move on from that one because, as I said, it's a dying platform, in my opinion, at least anyway. Where is my cursor? There we go. Now, so that previous one was the only example of sequencing by ligation that you really have to worry about. Okay, so just remember, because it says sequencing via ligation, it's a ligase that's being used, and it's not actually, the sequencing component of it isn't actually relying upon a polymerase. The ones we're going to look at now rely upon a DNA or a polymerase. Okay? For for the sequencing component of it. So these are the sequencing by synthesis, so SBS. The first form is we're going to do these cyclic reversible termination ones, so the CRT ones, and these are the ones that utilize bases that are terminally blocked. Okay, so this description that we've got on the screen here is Illumina. Illumina is the company that has the greatest market share in next generation platforms. So much so that it's almost monopolizing the industry. Okay. So what we get here is it relies upon, for if you remember the way the clonal amplification strategies, it relies upon those bridge-based synthesis, bridge-based amplification reactions. So we end up with the clusters on an attached substrate, and then what happens is we have a bunch of the different terminated but fluorescently labeled nucleotides come in and obviously bind and release a signal, and then what happens, the signal is cleaved, washed away, and the terminating 3' hydroxyl group is regenerated so that it can allow a polymerase to add on another base. Okay, it's reasonably simplistic to understand. So if we go through that again, when we have our clonally amplified templates attached to the substrate, flooded with the four labeled but terminally blocked nucleotides. They'll bind to their complementary sequence. You can't have another one added on. Why? Because I said they were blocked at the 3' hydroxyl group. So the polymerase can only add one of them on. So it can only put the one on at a time here. Okay. Then the fluorescence is measured. We can detect that with a CCD camera, like you have in a digital camera, and then the fluorophore is cleaved, washed away, and the terminating 3' hydroxyl group on that base is regenerated, which means it's put back so that it can actually allow another base to be placed on, and it cycles through again, and eventually you get all the different bases added on, and each time they're added on, the fluorophore is released. Okay. Then we've got this single nucleotide addition method. So we start off with our pyrosequencing, which belongs to the 454 line of sequences made by Roche. Here, it's a little bit of a different technology. You can only be reading the one type of fluorescently labeled nucleotide at a time. Okay, so in this particular method, we have a bead that has all of our clonally amplified fragments on it. Okay, so here's one of them extended. What happens is we have a base attach. Okay, the polymerase will go through, and if that base, I suppose I should add here that only one base is flooded over the bead. Okay, so the bead is usually placed into a well or attached to a plate, and then the plate or the well is flooded with only one nucleotide at a time. If it happens to bind, okay, then it will cause a reaction as the polymerase extends and it extends over it, and it releases inorganic pyrophosphate. Now, all we need to know now is that that inorganic pyrophosphate reacts with an enzyme that is also in the mix, and it triggers a cascade that ends up in having light produced by this luciferase enzyme. Okay, so light's produced whenever something binds. Let's say that there was an A on our sequence and we flooded it with a T. We'd have the binding, which would trigger the cascade, and we'd get the light produced, which then the little camera detects. Right? If we flooded it with a G, there'd be no binding, no light. It's there, all those G's are then washed off. There's nothing recorded, so we know it's not a G, and you'll only get the recorded signal once the actual appropriate complementary base is flooded over the slide. Okay, so that's single nucleotide addition. So each base is washed over in an iterative process sequentially. Now the problems with this technology, they can give quite long reads, up to 700 bases for the 454, which is approaching some of the Sanger type technologies, but they really struggle when you have multiple of the same nucleotide. So say we have an A, but the next one also requires an A. The polymerase will put both of them on there. Okay, it can probably do both quite well, and what happens is you get twice the amount of pyrophosphate, so you get twice the signal. It can read that, but it struggles when you start to get up around 6. So if it's had to add 6 in a row, like 6 A's, because the A's being flooded and there are complementary positions for the A's, it can sometimes misread how many. It might say there are 5 or 7 because you're trying to differentiate the difference in light intensity produced by that cascade reaction. So that's its disadvantage, okay, with those what we call homopolymer nucleotide runs, so runs of the same nucleotide. It is, but obviously there's all different sensitivities of the machines, and it does a pretty good job, but when you're doing it over countless thousands and thousands of reactions, you only have to have a mistake every now and then for it to become a problem. Okay, so we won't bother looking at a little, so that's called pyrosequencing, and it's basically because of the light emission. Okay, so that's a part of the sequencing by synthesis. So we're using a polymerase and we're having single nucleotide addition. So you're only having one nucleotide introduced to it at a time. There's another type of single nucleotide addition, and this one's used by ion torrent machines. This is the only machine of those short read sequences that are commonly used that doesn't use a CCD camera. Okay? It doesn't use a little camera to detect any fluorophore or light emission. What it actually does is it detects pH changes, so it detects a pH change when a hydrogen is released, and whenever the polymerase extends a base, a hydrogen is actually released. And there's a we can think of it as the world's smallest pH meter attached to the plate, and it can record those changes. And it's very similar to the pyrosequencing in that if we take a bead that's been clonally amplified and we wash over, let's say here, a load of A's, the A will match, is complementary to the T here. So the polymerase will extend. When it extends, a hydrogen ion is released. So before we had a pyrophosphate which triggered an enzyme cascade resulting in light. This time we have the hydrogen ion released, which can then be detected by a very small semiconductor. Okay, and we'll know that if we flooded it with A's, and we got a signal, then that's the position for an A. And likewise, if there were multiple A's required there, you get twice the voltage because the pH meter is being converted to a voltage. Okay, but it suffers the same problem with being able to differentiate long runs, long homopolymer runs. So if you have too many nucleotides being added, it can sometimes have problems differentiating between them. Okay, so here is an example of some different, and it says current NGS platforms, and this is what was on the existing slide. I had a bit of a look through it and I went, hang on a second, things have kind of progressed a little bit since then. So I then stole some tables from a recent review, and if we do a comparison between some of the platforms, and I've just thrown in here the solid, so this is the only sequencing by ligation short read platform that I've included. We can have a look at some of the read lengths, so we see they're quite small. This SE just means it's single end. I won't bother to go into single and paired end, but it's a very small 50 base pair read. And if we think about the throughput, we're getting 80 gigabases here. Okay. Number of reads, approximately 700 million. And what we haven't talked about is the time that these platforms take. So this, to generate that many reads, would take 6 days. So that's the machine sitting there for 6 days doing things. As we mentioned, it's very accurate. It's got a very small error profile because of that, the fact that the same template is being interrogated time and time again by those different universal primers that are offset. Okay, instrument costs, well, like all the cost of these instruments, when they start out, they can be very expensive, like up to $1 million. Okay, and then they usually drop in price. Some of them come down to a price that is perhaps affordable by a laboratory, and maybe in the order of 50 to 250,000 or something. But a lot of the newer, larger machines are highly expensive and therefore out of the reach of most laboratories. Cost per gigabase, also kind of an interesting one to look at. We can see here it's $130. Now keep that in mind when we come down and have a look at some of the other technologies. So sequencing by synthesis using the cyclic reversible terminators here, termination technique. This is the main platform here. This is the main technology being used. Illumina, as I said, they're the company that basically dominate the market. Okay? And they have a lot of different machines out. They have machines that they've flooded the market for, so you can have something that would be as close to having a desktop sequencer as possible, right the way through to what is called the HiSec X, which you can only purchase from Illumina in groups of 5 or 10, and each one of them is about $1 million. So you can see that only large companies that specialize in sequencing would actually be able to afford them. But they're the ones that can pump through like 10 human genomes in a run at 30 times coverage. So they're designed for mass sequencing. But there are smaller machines, and you can see here the read length is somewhere usually in the range of maybe 150 to 300 reads. We've got the throughput, you can see it goes right up. The number of reads is quite high. The error rates are quite low. The time, well, some of these smaller ones can be as low as like 7 hours, maybe 4 hours here if you're not getting a high throughput. But then we still end up in the days if you're trying to get something with any great throughput through. So if you have a large amount to actually read, then it's going to take time. And then right at the bottom here, we can make our way through. These are just basically the larger and larger machines. Here's the Illumina HiSec 10, the one that's specifically designed at amplifying basically genomes and lots of them. The one that you can only purchase 5 or 10 at a time from the company at about $1 million each, and it pumps out a lot of data. We're talking 2.6 to 3 billion bases. Okay, and then you're talking days once again for it to do it, to finish its run, but you get a lot of data. Okay, don't worry too much about this. I think this is meant to be in thousands, thousand, so it's like a million. Then there's some single nucleotide addition sequencing by synthesis here, and we have these 454s, which are the Roche. Okay, so these are pyrosequencing, the one that emits the light. And we can see that they've got a slightly higher insertion deletion occurrence. So if you want to put it, they're a little bit less accurate than the others. They're usually quite a lot cheaper, and they kind of range from a low to, let's say, a medium output. Then we have this ion system, and that the ion torrent systems are the ones that are measuring the hydrogen release, so the pH changes. So still doing the single nucleotide addition, and these are the most affordable for a laboratory. The prices for the machines are substantially lower. This is like 49,000 or so. Some of them are a little bit higher, but their modern Ion S5, which was released last year, I think is about 65,000 US, and it can put out a lot of data, and it's made as what they call plug and play, as about as user friendly as you can possibly get. So the machine sits on a space about the size of a small table, and you get everything as basically a clip-in adapter. So think of it how you change cartridges in your inkjet printer. Basically everything gets supplied to you like that by the company. So you just clip everything in, wash buckets, wash solutions, nucleotide cartridges, everything is just plugged in. Your template preparation is done on a separate machine called the Ion Chef, and it basically automates everything as well. So in as short a time as possible, you get all of your DNA put onto a little tiny semiconductor chip. Once you've got it on that semiconductor chip, you just plug it into the large sequencer and walk away, and it does it all, okay, at a cost. But a very fancy machine. And then, we haven't spoken about them yet, we have some of the long read machines, which I suppose we could still, which are really still in a high development. The others, although they're still quite new, have some history, so they're kind of true, tried and tested. The single molecule long read machines are still developing. They still have reasonably high error rates, and we'll discuss those in a minute. And then there's some very new technologies, such as the Oxford Nanopore, which take a whole different approach, and we can consider those future technologies. It says approximately, that's the approximate symbol. So that's approximate 12% error, so that's huge, especially if we're used to looking at Sanger stuff at 99.9999% error rate, and then you come to something that has a 12% error rate. You can see that that's a developing technology, but when we discuss it, you'll see that it promises so much that if that can be reduced, it's a game changer. Okay, the PacBio systems, which we also would discuss, a long read machine. It's got 13% kind of error rate here. Now it uses all sorts of fancy methods to correct for its error, and apparently says can be 99.999% if you bother to go through all of these different methods to reduce its error rate, usually through computer algorithms, etc. Alright, so we'll get on to these long read sequencing platforms, which basically we can consider third generation platforms, third generation sequencing machines. So long read sequencing. So you might see single molecule real-time sequencing thrown about, so SMRT. Here you're not reliant upon an amplification step. So I've broken down the technologies to short read and the long read. all of the short read technologies are requiring an amplification process. Okay, so fragmenting, fragmenting, ligating on adapters, clonally amplifying, then going through those sequencing technologies. The long reads, you're not reliant upon PCR amplification, and because of that, you're not as reliant, or you're not as susceptible to biases that could happen, because like Chinese whispers, every time something's amplified, you increase the chance of having an error. Okay, so you're looking at its real state. It's probably best to, and I thought I had a little clip here. The reads from these machines, these long read sequences, and the most popular one being one developed by a company called PacBio, they've actually recently released a machine called the Sequel, which if we flick back to here, you can see we're talking about 8 to 12,000 bases in a read. Now if we have a look back, the solid system, we were complaining about it having 35 bases, and then the leading instrument at the moment, the one that most people use that monopolizes the system by Illumina, we're looking at a maximum of 300 bases for each read. You can see why these are called long reads. Okay, so now we're at 12,000 bases, and in their previous model we're talking like 20,000 bases. A lot of data coming out, but a lot of long reads. Now, why are long reads great? Because you don't have to then piece together a jigsaw puzzle. As I say, I was asking you to put together a jigsaw puzzle so you could identify the picture in the jigsaw puzzle. A lot harder if you have lots and lots of pieces than if you have just two pieces. Okay, so there are definitely advantages to having long reads. Interestingly, I was mentioning some of these machines are kind of now obsolete. This Helicos biosciences system here, which was similar to Illumina sequencing, is now bankrupt. And my favorite, the little 454 Roche, which they still are selling, is basically now what I consider a zombie. It's Walking Dead. It's Roche have basically said they're no longer developing it. So as of 2016, they said that they're still heavily advertising it on their website, etc. But it's pretty much going to go the way of the dodo pretty soon. So that Roche 454 was the one, and probably the only machine using pyrosequencing. So we may no longer see pyrosequencing in the next 5 or so years as a technology. So getting back to the long read systems, the PacBio, lots of reads and lots of long reads with unfortunately a not so great error rate. Okay, as I said, you can kind of overcome it with some mathematical gymnastics, but this error rate is slowly decreasing, and who knows, in the next 5 years, if this gets somewhere near the 99% region, then having long reads is just a wonderful technology. It's much easier to piece together a whole genome if you have large pieces than lots and lots and lots of little pieces. So we'll just quickly have a look at its little clip, just to show you how it works. It is a little bit harder.
Biosciences is developing a transformative DNA sequencing technology that will revolutionize the field of genetic analysis by enabling researchers to answer questions important to human healthcare. We call it SMART, Single Molecule Real-Time DNA Sequencing, a breakthrough technology based on the natural process that occurs every time living cells divide. Prior to division, DNA is replicated by enzymes called DNA polymerases, which efficiently duplicate entire genomes in minutes by reading the DNA and sequentially building a complementary strand with matching building blocks called nucleotides. Pacific Biosciences' smart sequencing harnesses the power of the polymerase as a sequencing engine by eavesdropping on it while it works to replicate DNA. This approach is enabled by two proprietary technologies. The first is phospholinked nucleotides. To visualize polymerase activity, a different colored fluorescent label is attached to each of the four nucleotides, A, C, G, and T. In contrast to other sequencing approaches, our phospholinked nucleotides carry their fluorescent label on the terminal phosphate rather than the base. Through this innovation, the enzyme cleaves away the fluorescent label as part of the incorporation process, leaving behind a completely natural strand of DNA. This enables us to exploit the inherent properties of the DNA polymerase, including high speed, long read length, and high fidelity. The second key technology is a nanophotonic visualization chamber called the Zero Mode Waveguide, or ZMW. It enables observation of individual molecules against the required background of labeled nucleotides, while maintaining high signal-to-noise. The ZMW is a cylindrical metallic chamber approximately 70 nanometers wide that is illuminated through its glass support, creating an extremely small detection volume, just 20 zeptoliters. Nucleotides diffuse in and out of the ZMW in microseconds. When the polymerase encounters the correct nucleotide, it takes several milliseconds to incorporate it, during which time its fluorescent label is excited, emitting light that's captured by a sensitive detector. After incorporation, the label is clipped off and diffuses away. The whole process repeats, creating sequential bursts of light corresponding to the different nucleotides. These are recorded, thus building the DNA sequence. Our smart sequencing harnesses the polymerase's natural ability to synthesize 10 or more bases per second over thousands of continuous incorporations, leading to high-speed and long-read sequencing. Additionally, our smart technology design allows simultaneous multiplexing of thousands of ZMWs in parallel.
Okay, so that's basically what we needed to know about that. So you have the polymerase, which is attached in that ZMW little microchamber, and in that, as you saw, as the right nucleotide is attached through the polymerase working, it gives off that light. Okay, so quite a different approach, but it allows those really long reads, but unfortunately at a little bit of a higher error rate. Well, it's just basically the technology itself. It's obviously not a. I mean, if you think about it, what was he talking about? 10 to the minus 21 liters or whatever, which is a very, very small volume. And then you're trying to read a fluorescence that's being emitted by the cleaving of a polymerase that's working at more than 20 or so bases a second. It's just blurs the lines of how well it can detect that. The polymerase is moving so fast, there's lots of bases being added and potential fluorescences occurring. It's an inherent error rate due to the speed and the ability not to differentiate the different fluorophores there at any one time. But, I mean, the technology is improving. It's still an incredible technology nonetheless. Okay. So then there's this last one, this Oxford nanopore, and this one, if it actually comes to fruition, is a true game changer. We've got number one, this mini ion, and I'll go through the technology in a minute, but this mini ion is a sequencer, and that's a USB plug there. That's someone's hand. So you can plug that in, put your sample in it, and it can do the sequencing then and there. Okay, you have to plug it into a computer. They've also got a smidge ion, which is an adapter for your phone. Okay, so this one's brand new release. Plug it into your phone, add a sample onto it, and it can give extremely long reads, like long reads. Now, it, like the other long read machines, suffers some error rates, up to 15%, but if you're only sequencing very small things, then those error rates aren't so much of a problem. So this little mini ion has been successfully used through epidemics and disease outbreaks, like the Ebola outbreak a couple of years ago, to actually sequence the Ebola genome in real time. Probably only took about 15 minutes or more to get it sequenced, but you've got it then and there. In the field, applications are huge. Now, even with the high error rate, 15%, they can overcome this by having, basically, this is the Promethean. So basically lots and lots of these. So you can just do it so many times that 15% error rate can be mathematically taken out of the equation. So you can get very reliable estimates of what you're actually looking at. Now, it works on a very different technology, and hopefully, oh no, that's not what I wanted to look at. Bear with me a second. How it works. So all those machines listed there work on fundamentally this same process, which you click on here, hopefully
Oxford Nanopore Technologies is developing nanopore-based sensing technologies for the analysis of biological molecules like DNA, RNA, or proteins. At the heart of the technology is a protein nanopore. This model shows a typical nanopore. You can see that at the core of the protein is a hollow tube that is only a few nanometers in diameter. Oxford Nanopore designs and manufactures bespoke nanopore structures for a range of applications. In nature, nanopores form holes in membranes. In Oxford Nanopore's technology, the nanopore is inserted into a membrane created by a synthetic polymer. This membrane has very high electrical resistance. By applying a potential across the membrane, which is bathed in electrochemical solution, an ionic current can be generated through the nanopore. Single molecules that enter the nanopore cause characteristic disruptions in the current. This is known as the nanopore signal. By measuring that disruption, the molecule can be identified. Larger target molecules may pass near the opening of the nanopore rather than passing through it. This might, for example, be a protein molecule. This can also create a distinctive disruption in the current, which can be measured through the open nanopore. Using microchip fabrication techniques, Oxford Nanopore has developed devices that enable highly scalable arrays of these nanopores to be used for real-time sensing of biological molecules. The number of nanopores used can be scaled up to industrial levels, depending on the application and the turnaround time required. For more information on how this technology is being used by Oxford Nanopore, please visit Nanopore.
So you can see that works on a totally different methodology where you're basically having DNA just passing through that nanopore and it recognizing each of the base. So just like a ribbon being fed through, and it just is endless. So and that's why they can be miniaturized into these small devices. Now I spot here it's semi-release, so this mini ion, it's not really commercially released. So the company allows people to buy them and have them, but they always have to be in close contact with the company, reporting and kind of helping to improve the technology as it goes. All right. We spoke about the cost of some of these machines. Some of them are really restrictive for laboratories, and things like the Illumina HiSec 10, only large sequencing companies could afford to have them. And there are several large sequencing companies that well, that's all they do, that's all they specialize in, so they have all these different sequencing platforms available, and you just send your samples off to them. And that's how we work in our laboratory here. We can't afford to purchase $1 million or $500,000 machine, nor could we afford to actually maintain it. So it's much easier to send off to a sequencing servicing center to do it, just as your samples were sent off to AGRF, so Australian Genome Research Facility, for just simple Sanger sequencing. Okay. So what do you really need to know? It'd be great if you could at least get the fundamentals of the next gen sequencing, understand that it's massively parallel, that there are huge amounts of data involved with that. I think in 2013 calculation was that something in the order of 15 petabytes could be generated, and that's gone through the roof. Now it's even greater. So there's a massive problem, downstream problem with bioinformatics and how you deal with this much data. So not only do you have sequencing centers, but you also have these large computing facilities and cloud storage facilities that are required to hold your data and actually the computing power to manipulate the data. Okay, the reduction in the cost of sequencing is just plummeting. Okay, using our Sanger sequencing, we can really only afford to do it for a few sequences. It's $4 a sequence at its cheapest, which if you were able to scale down the next gen to actually do our samples, then it would be a matter of like maybe 4 cents or less. So the only reason we use it is because it's high accuracy and we can kind of afford it at such a small scale. For large projects, then NGS is the only way to go. You should know that the difference between the synthesis by ligase and synthesis by sequencing, okay, one using ligase, the other using polymerase. It'd be great if you could understand the 4 common steps, so the fragmenting of the DNA template, the adaptation of the ligating of adapters, the clonal amplification, and then the sequencing, and be able to describe some of those sequencing technologies. Okay. The single molecule stuff for the long reads, great if you can understand those, but they're not commonly as used, so you'd be much, or I'd advise you to, if you're going to put the time and effort into understand the short read stuff more so. Okay. So that last little point there is the data collection and time being a problem. Cost is not so much of a problem these days. Processing of this data, once again, needing fast computing power, especially from short reads. Short reads need to be assembled. This is the problem with them. And that assembly is very computer intensive. It's like a big jigsaw puzzle. And it's okay if you have something to match it up against, but if you're just trying to figure out through computer techniques and mathematics a whole heap of little strands of what possibly matches with what without having something to compare it to, it can be very difficult when you have lots and lots of smaller pieces. The larger the pieces, it's much easier to put together jigsaws with large pieces. So here, just some, what we call, and putting those together is called assemblies. So here we have some assemblies of some different reads. This is done in Genius. We no longer use Genius, so it's probably a bit harder to understand. But fundamentally, if we're thinking about aligning something, you've sequenced a human, let's say we've sequenced your genome and you've broken it up into lots and lots of small little pieces. If we have a reference genome, so we already have the Human Genome Project which has been completed, we roughly have a map. We know where things should kind of go. So your genome isn't going to be that different to the one that's already out there, so we'll know what little pieces should go where, and then we can identify any little differences. So that's what I mean by putting the jigsaw together, but much easier with longer reads. But the trade-off is accuracy at the moment as we stand with the technologies. And there we go. I advise you to make the most, if you struggle with my interpretations of those, to have a look at some of the little video clips on the different technologies. Some of them, like all of these animations, etc., are easier to understand than others, and some of them are just full of propaganda. Okay, and I'll see you this afternoon.